All right, we're going to get started. Last time we started covering the mechanisms of transcription. We learned about consensus sequences and why those were important in regulating transcription. We learned about the first step of transcription, which is initiation. Quick review of what initiation was in transcription. Initiation is the whole act of getting RNA polymerase ready to transcribe everything. The way this works is RNA polymerase has this extra little component called the sigma subunit, whose primary job is to bind to a promoter sequence on the template DNA. In doing so, you form what is called a closed promoter complex, which is what happens when RNA polymerase initially binds to its target DNA. And then through the initiation process, DNA opens up. When DNA opens up, you form an open promoter complex. Pretty easy to memorize that. And then the first couple nucleotides are added to form the very start of your mRNA in transcription. However, after the first few nucleotides are added, your RNA polymerase is stuck. In order to finish the initiation process, the sigma subunit needs to leave. So that's the definition of when initiation ends and elongation begins, is when the sigma subunit leaves. When the sigma subunit leaves, you remove the parking brake. RNA polymerase then moves along the template DNA to transcribe the rest of the mRNA. That whole process of transcribing the rest of it is called elongation, of which there's very few details you need to know about elongation. Let me go to this slide here. Because all elongation is an extension of the mRNA strand. Some interesting facts would be, one, RNA polymerase doesn't have an editing or proofreading mechanism like DNA polymerase did. RNA polymerase isn't able to correct itself, so it regularly makes mistakes. When it makes mistakes during transcription, usually that mRNA will get thrown away later. Other important facts regarding elongation is that when you unwind DNA in order to transcribe it, you create supercoiling elsewhere. You create positive supercoiling ahead of this transcription bubble and negative supercoiling behind it. And we know we have enzymes that exist to fix these things. We know gyrase is going to fix positive supercoiling, and we know general topoisomerases will fix the negative supercoiling. We started to talk about how transcription ends, the last step in transcription, which is called termination. And in prokaryotes, this is only a prokaryote thing, there are two ways transcription ends. One is called row-dependent termination, where transcription ends because of a protein called row. And another one is called row-independent termination, where it ends with some other method, which we'll talk about today. So to review how row-dependent termination works, this slide is a nice visual of it. Basically, there is a protein called row. It binds to a site on the mRNA strand called the root site, I don't know how it's pronounced. It binds to a particular site on the mRNA strand and then travels up along the mRNA strand until it catches up to the transcription bubble. When it catches up to the transcription bubble, it uses its helicase activity. The row protein, it turns out, has a special helicase activity whose job is to unwind everything from each other at the transcription bubble. And when you unwind the DNA from the RNA and you unwind the RNA polymerase from everything, you can't transcribe anymore. And this is how row-dependent termination of transcription occurs. So that one's actually pretty straightforward. Let's see, let me find a good picture. I think this is it. Let's see. This one's all right. So, how does row-independent termination work? Row-independent termination does not require the protein row to end transcription. Instead, the sequences itself will end transcription. And what do I mean by that? Well, remember back in like 10b or chapter 11, I think, we learned about these specific DNA patterns called inverted repeats, right? Back there, we learned that they formed cruciform structures, right? Over here, in the context of transcription, inverted repeats will form these shapes here. hairpin loops. If you transcribe an inverted repeat, the mRNA will take the shape of this hairpin loop, okay? Not a cruciform, because a cruciform is a double-stranded thing. When in transcription we talk about that it forms this hairpin loop. This hairpin loop, it turns out, is a very destabilizing structure, especially in the context of transcription, where termination ends when a hairpin loop forms, in addition with all these A's and U's on the tail end. So the way, and I'll say this again, so the way transcription ends via row independent termination is that a transcription of an inverted repeat occurs, in addition to there being lots of A's and U's association at the end there. Why is that important? Well, G's and C's are made of 3 hydrogen bonds. A's and T's, which is the same thing as A's and U's, are bound together via 2 hydrogen bonds. So, if you, 1, create this hairpin loop structure, which is a destabilizing structure, and 2, you're only hanging on barely by a uracil adenosine association, hydrogen bond, those things put together allow the mRNA to simply fall off the DNA template, okay? So row independent termination is this hairpin loop is formed because you've transcribed an inverted repeat, and all these A's and U's on the end there are not very strong in terms of hydrogen bonds, so everything just falls off. All right, this is row independent termination. Maybe I oversimplified it, but you can look at the notes here for a super detailed explanation of it, but that's how I keep the 2 types of termination distinguished from each other. Let's see if there's anything else with that. Again, I will stress that this is a prokaryote thing, not a eukaryote thing. Let's see. Oh yeah, and this talks about, by the way, what are some consequences over the fact that RNA polymerase does not have the ability to correct mistakes. Most of the time, when RNA polymerase makes a mistake during transcription, the mRNA is useless. It doesn't really get translated into anything. This slide will talk about how sometimes, even though the RNA polymerase makes a mistake during transcription, it creates an mRNA that can still be translated into a protein, a unique protein, one with a slight diversity than what it was supposed to be made into. And you can go ahead and look at the details of that here. Other than that, let's see, transcriptional regulation. Oh, okay, so it's time for the Lac Operon. All right, so for those who've taken a genetics class, and sometimes they even go over this in microbio, you learn about operons, Lac Operon, Tryptophan Operon, stuff like that. In this class, we really are only going to focus on the Lac Operon because it is a good explanation of how transcription is regulated in prokaryotes. I'll stress again, the Lac Operon is a prokaryote thing. The way it is regulated is going to be completely different than how transcription is regulated in eukaryotes. So that is a common mistake I see people mix up with on the exam. They think the Lac Operon is found in both prokaryotes and eukaryotes, and all the stuff that we're about to go into, they get mixed up with. So before we get into Lac Operon, let me make sure I have the slide I want to go to. 1 second, just ignore all these things. I will cover them in a second. All right, let me make a note here. All right. So, before we get into the Lac Operon, the way transcription is regulated in prokaryotes is via 4 possible combinations of terms that we're going to see. You're going to see in a bit that when they describe regulation of transcription in prokaryotes, they use either a combination of positive versus negative, so they'll either use the positive term or negative term. They'll say either positive control or negative control. And here's what each one means. Positive control means that whatever happens, it requires an inducer protein. Okay? In order for positive control of transcription to occur, an inducer protein is involved. The phrase positive control does not automatically mean that transcription will be increased or decreased. It does not mean that. That's a common mistake people think. Oh, positive control must mean transcription is going to happen faster. No, technically the positive control only means that a protein whose job is to induce something is involved in this control. And I'll give you examples in a bit. Negative control means a repressor protein is involved in some way. It does not mean transcription will be decreased. That's not what negative control means. Negative control means that a repressor protein is involved in some way, and we will see how. After you go through those terms, there are two other terms here. These are probably what you are more familiar with. If something is being induced, if transcription is being induced, meaning induction, transcription is activated. Is activated or increased. Maybe there was some transcription going on. Induction means even more will happen. So transcription is activated or increased. Repression also is more, logically sensible here. Repression means transcription is deactivated. Deactivated. Or if there was transcription going on, repression, rather than just deactivating it, could decrease transcription. Maybe not completely shut it off, but at least decrease it. Okay. Yes, these words are only associated with prokaryotes. In eukaryotes, you'll learn about things called transcription factors, enhancers, proximal and distal promoters. You'll learn all about that later. This is just for prokaryotes. All of these terms, you can create 4 combinations here. You can create a combination that is positive inducible or positive repressible. That sounds weird, right? Negative inducible sounds weird. Negative repressible. But all 4 are technically possible, and different systems, different operons will be regulated via one or more of these ways, of which we'll go over in a second. But this you should internalize. Back when I was undergrad studying microbiogenetics, biochem, it took like the 3rd time before I could figure out what these things meant. They don't usually do a good job of explaining the difference between positive control and induction. They are very different things. The phrase positive control means a protein whose job is to induce things is involved, but the phrase induction actually means transcription will be activated. The end result is transcription is activated. And similar definitions over there. So, before we get into that, let's talk about what the Lac Operon is and then what regulation it undergoes. Okay. Let's see. So, remember how I said in prokaryotes they are polycistronic, right? What that meant is that several genes will be transcribed at the same time, because in prokaryotes, genes are right next to each other in the genome. They will all be transcribed at the same time. An example of that is the Lac Operon. The Lac Operon is made with, 5 genes. The Lac Operon is made of 5 genes. These genes will all be transcribed at the same time, and they are all regulated at the same time via a region called an operator. Okay? That operator, we will see later, is where inducers can bind to, repressors can bind to, things can go on it and off it, and whatever combination of that, we will see in a bit, either will turn on transcription of the Lac Operon or turn off transcription of the Lac Operon. So, what do you need to know? Well, the whole purpose of the Lac Operon, by the way, and I guess I should start with this. This is sometimes your extra credit question on exam 3, is to describe how the Lac Operon is regulated. What is the Lac Operon composed of? What is the purpose of the Lac Operon? How is it regulated? There's technically 2 ways it is regulated, and you need to talk about both, and we'll go over that. And give me that information. Show me that you know what you're talking about. I'll give you the full credit. All right. So, What is the goal? What is the purpose of the Lac Operon? Well, prokaryotes want to break down lactose sometime, but in order to do that, they need to create a special enzyme to break it down. That enzyme is composed of the multiple genes that you see in the Lac Operon. You don't have to know anything too crazy about it, just that the purpose of the Lac Operon is to create a protein to break down lactose and metabolize lactose. Okay? I'm going to skip all those structural things because you don't need to know them. It's a little bit out of order, but then they talk about how one of the genes in the Lac Operon itself is the repressor. I'll talk about the repressor in a bit, but they're talking a little bit about the different genes that you find in the Lac Operon. Feel free to look at this if you want. It's not necessarily as important as understanding the regulation of the Lac Operon first. And then you can go back to this and read this slide. So I'm just going to skim through these things. Oh yeah, if you want to know what each gene is called in the Lac Operon, the Lac-I gene is called the repressor. The Lac-Z gene is called beta-galactosidase. It sounds like its job is to break the beta linkage in the galactose glucose bond in lactose. Permease and transacetylases, they are involved in like transport, I don't remember exactly what, and you don't have to know exactly what. But these are the names of the enzymes that are composed of the Lac Operon. All right, so are we at the regulation part yet? Okay, we are. So instead, I'm going to draw a simplified version of this. All right. So, the Lac Operon, out of all those combinations of words, the Lac Operon is regulated via negative inducible control. Okay? Negative inducible. All right? That's how the Lac Operon is initially regulated. This is step one, and it's important to talk about step one because technically there are two ways the Lac Operon is regulated, but this has to happen first. Let's break it down. Let's break down the inducible word first because that one's more logical and easier to understand from the get-go. What does inducible mean? You're going to turn on transcription. Therefore, the Lac Operon is initially turned off. Okay? The Lac Operon is initially turned off. And the phrase inducible means your goal here, the way this will be regulated, is it needs to be turned on. Other types of operons of which you don't need to know are called something repressible. That means normally whatever that operon might be is constantly being transcribed over and over again. And when it is regulated, it needs to be turned off. This one, because it's inducible, it is initially off and needs to be turned on. A good reason why that is because, well, lactose isn't necessarily the most abundant sugar that bacteria wants to metabolize. Bacteria love glucose. They really will only start breaking down lactose if there's no choice. So that's why they want to keep this operon off. Otherwise, what's the point in transcribing the genes of the Lac Operon? You want to keep it off until you need to break down lactose. So that's why it is initially off and you only turn it on when you need it. All right, the phrase negative means that in some way a repressor protein is involved. Okay, a repressor protein is involved. How can something be negative inducible? How could a repressor protein be involved in turning something on? And here's how. Initially, here's the Lac Operon. And here's the operator part. Initially, there is a repressor protein on it. So initially, a repressor protein is on the Lac Operon, preventing transcription of the Lac Operon. The repressor protein is on the operator here. There is no or very little transcription. All right. What's going to happen is that there will be a co-activator Or is it co-inducer? Let me make sure because I might be getting the eukaryotic term mixed up. Let me make sure I don't give you the technically wrong term here. It is co-inducer. Yeah, I used the wrong term there. Co-inducer. Okay. So there's going to be something called a co-inducer whose job is to remove the repressor from here. Okay? That's the inducible part. The inducible part is your job is to try to turn on transcription. Well, logically speaking, if you want to turn on transcription of the Lac Operon, your goal has now become to remove the repressor from this thing. The molecule that will do this is called a co-inducer. The co-inducer's job, this is negative inducible. The co-inducer's job is to bind to the repressor, changes something about the repressor's structure, and then the repressor just falls off the Lac Operon. So that what you have now is free and open Lac Operon. The repressor protein is maybe somewhere down here off to the side with the co-inducer bound to it, preventing it from rebinding to the Lac Operon. The co-inducer's job is to remove the repressor protein and keep it away from the Lac Operon. Now that the Lac Operon is open, transcription can occur, all right? Well, it's technically more complicated than that. What I mean that transcription is occurring, I'll go back to it after I go over the second way this is regulated. But to summarize, negative inducible control is how the Lac Operon is regulated. This means that there is initially a repressor protein on there, and you need to remove it. And the thing that will do that is called a co-inducer molecule. For the Lac Operon, the co-inducer is lactose, or a slight modification to lactose called allolactose. And there's more, but these are the two that are most talked about. This makes sense, right? If lactose is present, wouldn't you want to turn on this thing? Yes, so that's why lactose itself will actually remove the repressor off of the Lac Operon, okay? And again, I am oversimplifying it, but you don't need to know the specific details of this whole lactose, allolactose thing. Just the general thing is that these molecules, when they are present, they will remove the repressor protein off the Lac Operon. Okay, so these act as the co-inducers here. All right, so the end result is you have turned on transcription of the Lac Operon. Pretty easy so far. Okay, any questions? That's half of it. We got to go to the other half now. All right. Now, although you've turned on transcription, in reality, it's actually quite slow. All right, you've turned on transcription because you removed the repressor. An analogy of this is you've removed the parking brake on your car. It might drift forward a little bit, but it's not going that fast. This term is called leaky transcription. When you've turned on transcription technically, but it ain't going that fast. Okay? So, part 2 of the way the Lac Operon is regulated, the goal of that is to really ramp up transcription. So, let me jump to that. Okay. Part 2 involves a protein called the catabolite activating protein. I just say cap protein. I think it's called catabolite activating protein. You can look for the specific name in your notes. I will accept cap protein. This thing is part 2 of the Lac Operon regulation. It is involved in positive inducible. Yes? This is, for the Lac Operon, if it ends up being the extra credit, you need to describe both of these things, both the first part and the second part. You have to explain both of them. All right. Because they're both necessary for regulation of the Lac Operon. So positive inducible, is the regulatory phrase we use to describe the cap protein? Well, let's break it down. Inducible, pretty easy. Either you turn on transcription or you really increase transcription. So I'm going to put increase here because we know transcription's already on. So our goal is to increase it even more. The phrase positive means an inducer protein is involved in some way. And specifically, for the Lac Operon, for part 2 of the Lac Operon, the inducer protein actually has a name this time. It is the cap protein. The catabolite activator protein is your inducer protein this time. Okay, so there is a name for it. You need to actually give the name for it if you're describing how part 2 of the Lac Operon is regulated. All right, so now let's draw some stuff. So, initially, so after part 1, here's what we have. Our free operator and the Lac Operon, which are all the genes of the Lac Operon there. Okay, it's open, you've removed the repressor. But now, you want to attach the cap inducer. You want to attach it to the operator, because in doing so, it's going to really ramp up transcription of the Lac Operon. You are going to, if this thing attaches to the operator, it's like stepping on the gas in your car that you have removed the parking brake from. So, how does it do that? Well, it turns out there are also co-inducers for this thing. So there are also co-inducers for this thing that will bind to the cap protein, change its structure slightly so that it prefers to bind to the open operator. All right, so that's what the co-inducers here will do. These are not the same co-inducers as step 1. Step 1, what were the co-inducers? Lactose or allolactose, right? Or anything that kind of looks like lactose. For step 2, we have different co-inducers for the cap protein. Those are cyclic AMP. I think it's the only one you need to know for this. So cyclic AMP are your co-inducers for the cap protein. All right? So if cyclic AMP is present, it will bind to cap, change the structure of the cap inducer protein a little bit so that it prefers to bind to the empty operator now. And when it binds to it, now, now that cap is bound to it, transcription is going to increase in respect to the Lac Operon. You really, I don't know if super activated is the exact word, but transcription is really fast. Lots of transcription. Let's see. All right, so that's step 2. Yes?
So then if you don't have the cyclic AMP, the cap will not bind to operator.
Exactly. Yep, and we'll get into that. That's the last part of the Lac Operon stuff we need to talk about, and you would have to describe on your extra credit is, what are the different combinations of step 1 and step 2 that you can have? You brought up a good point. Well, if there isn't any cyclic AMP present, cap won't bind to the operator, even though the repressor is gone. So then you're stuck with what we were at in step 1, which was you're just left with leaky transcription in that situation. We'll go over the different combinations in a second. But this, to summarize, is step 2 in the Lac Operon. The final step in the Lac Operon is the goal is to have the cap protein bind to the operator, and you ramp up transcription of the Lac Operon. You really do this when you need to break down lactose. So what are all the possible combinations that you have here? So we'll go over that now. So, step 1, or combination 1. Your bacteria is sitting in an environment, your prokaryote is sitting in an environment where there's lots of glucose, so I'm going to put an up arrow to indicate abundant glucose, and not a lot of lactose. Why did I mention glucose? Because I forgot to mention one last thing with step 2, so add this notes to your step 2 here. The levels of cyclic AMP is inversely proportional to how much glucose there is, meaning if there's a lot of glucose in your environment, this means there's not a lot of cyclic AMP. And if there isn't a lot of glucose in your environment, this means there's lots of cyclic AMP. Okay? A critical thing to know if you're studying for the MCAT is like the energy state in your cells. Why do I know that if there's not a lot of glucose, it means there's a lot of cyclic AMP? Well, remember, ATP, adenosine triphosphate, means that there's plenty of energy in the cell, right? If your cell instead has a lot of AMP, there's not a lot of energy in it. And you can trace that back to the reason being there's not a lot of glucose in it, all right? If you don't want to memorize that for this class, don't worry about memorizing it. I'm just kind of, if you're studying for the MCAT, one thing you should constantly be looking at, no matter what pathway they're talking about, is what is the energy state in the cell? Does it logically make sense? Yes, if there's not a lot of glucose, there's not a lot of energy being made. Therefore, there's probably going to be a lot of junk AMP floating around, not ATP floating around. All right. Anyways, you can choose to memorize that or just flat out memorize this inverse relationship for this class. Do whatever you want. But you need to know this in regards to part 2 of the lactose of the Lac Operon regulatory system. And that is, there can only be lots of cyclic AMP, there can only be a lot of this co-inducer if there isn't a lot of glucose. So, flipping back over here, if we have a lot of glucose floating around, this means there isn't a lot of cyclic AMP. And if you don't have a lot of lactose around, that means there isn't a lot of co-inducer for the Lac Operon. Let's illustrate this. Initially, way back in step 1, we have our operator, we have a repressor on it, we have the actual Lac Operon here. In this state, as I've drawn it, is transcription on or off? Transcription is off. If there is not a lot of lactose, or maybe there's 0 lactose, can you remove the repressor? No, because what was the co-inducer for the repressor? Remember that the co-inducer was supposed to be lactose, or some variation of lactose, all of which will be gone if there isn't any lactose in the first place. So if there is no lactose in the first place, you can't remove the repressor off the Lac Operon, so therefore, combination 1 means transcription is off. You can't transcribe anything. Okay? You cannot transcribe the Lac Operon simply because there is no lactose in the first place. It's really off. You can't be double off, but if you really want to look at it, if there's also lots of glucose, there isn't a lot of cyclic AMP. If there's not a lot of cyclic AMP, CAP cannot be activated either. All right? Honestly, I'm giving you this, but the CAP step does not matter in this case. Anyone want to know why? Can CAP bind to this operator if the repressor is there? No. So honestly, I could put question mark glucose. Who cares how much glucose there is? It could be lots of glucose or not a lot of glucose. The fact that there is no lactose is actually the reason why everything is off. All right? So in fact, we've come up with, in one step, we've come up with 2 combinations here. Combination 1, if there's a lot of glucose and not a lot of lactose, transcription is off. Combination 2, if there's very few glucose in addition to very few lactose, it is still off because step 1, the repressor is still on there. Does everyone get that? Is that if there's no lactose, you can't remove the repressor. It doesn't matter if there isn't a lot of glucose. Okay, you need to have lactose present. Okay. So those are 2 combinations so far. There's 2 others. Let's see. Let me make sure I do this in the right order. Okay. So, what happens if you have low glucose and abundant lactose? All right, well, step one. What happens in step one? If you have plenty of lactose, your repressor is gone. Good. Step two. If you have low glucose, what can you tell me about cyclic AMP? You have high cyclic AMP, which is great because that's what the inducer protein required. I will flip back to that right now. If you have lots of cyclic AMP, CAP loves that and will readily bind to the free and open promoter on the Lac Operon. So, if you don't have a lot of glucose, you have plenty of cyclic AMP, CAP superactivates the Lac Operon and really ramps up transcription, okay? So, the fact that you have low glucose and high lactose means that you have turned on transcription. You've really turned it on. Like, this is the maximum rate it could possibly go. Logically, this makes sense. Your bacteria is sitting in some environment. It's run out of glucose. It's desperate for food. It notices there's plenty of lactose hanging around, so it uses this two-step mechanism to begin metabolizing lactose. Okay, so always think about the energy state that an organism or these pathways are exposed to. All right, so far so good. Everything logically makes sense. But now for the fourth combination. And I'll start this on another page. What if you have a lot of glucose and a lot of lactose? Okay, well, whoops. What if you have a lot of glucose and a lot of lactose? Well, let's see. We draw our stuff again. In step one, since there's plenty of lactose, what happens to the repressor? It's removed, right? If there's plenty of lactose, repressor is gone. Step two. However, if there's lots of glucose, there is not a lot of cyclic AMP, so CAP can't bind to the Lac Operon. You have removed the parking brake, but you're not stepping on the gas. We found that there is a term used to describe that called leaky transcription. There is leaky transcription. So this happens when there is both glucose and lactose. The bacteria is happy. There's plenty of food for it everywhere of all varieties. However, it's picky. It prefers to use glucose instead of lactose. So, although the lactose caused the repressor to be removed, it's only transcribing the operon at a very slow rate, mostly because there's plenty of glucose for it. So this term to describe some transcription, but not the maximum rate of transcription, is leaky transcription. That occurs only when you have abundant glucose and lactose that the bacteria is exposed to. All right? So, stuff you need for the extra credit. Tell me, what does the Lac Operon do? What is the purpose of the Lac Operon? It is to create, it is to metabolize lactose. Lac Operon function. What are the genes needed for? It's to metabolize lactose. Two, let's see, 1, 2, I might be missing one. I'll go back over it later. 2 is tell me the way the Lac Operon is regulated, which is 2 parts, right? Tell me step 1 in detail, the components of it, the co-inducers, everything. Describe to me step 1, and then describe to me step 2. Which is the cap protein, the cap inducer. Tell me all about how you need cyclic AMP in order for the cap protein to bind to the open promoter. And 4, tell me all about the different combinations of material that you have here. What is the result? Remember, that is lots of glucose and not a lot of lactose. Tell me what the end result is. Every combination here, lots of glucose, lots of lactose. You don't have a lot of either one. And what combination do I have here? Very little glucose and plenty of lactose. Tell me what happens with these combinations here. And I think that's everything that I would require. Let me think real quick. What is the function? Tell me step 1, step 2. And especially in step 1 and step 2, you better tell me the different components. You got to tell me the inducer, you got to tell me the repressor, you got to tell me about what specific co-inducers there are. Don't just tell me co-inducer. You need to tell me lactose, or you need to tell me cyclic AMP. And tell me what happens in each of these combinations. It is worth 5 points, so I'll probably put more emphasis on these 2 parts here. But if you give me all that information, show me that you know what you're talking about, you'll get the full credit. Yes, question? The different combinations here, tell me basically, in this one, the repressor is still on there, and the cap protein, is not activated. End result is there's no transcription. Like 1, 2, 3. What happens with step 1? What happens with step 2? What's the end result for each of these combinations here? Yes?
So for what genes are needed for genes, what would the answer for that be?
For which one?
For what genes are needed? You said that part of the
Well, what did I say? What genes are needed? For these things, just what components there are, meaning the co-inducers in step 1 are lactose and allolactose. That's what I mean. For step 1? No, I used to require that you need to know every single gene, what every single thing does. No, just tell me what's the role of the Lac Operon. Like what is it needed for? We've gone over plenty. That really all its job is to make the enzymes necessary to metabolize lactose. That's the key phrase. That's the only reason we talk about this, is that you want to activate this thing so that you can metabolize lactose. Okay. So you can actually take this information, especially, let me go back to it, especially this table here, to help you break down any other operon you might run into. I think in other classes they go over another operon called the Tryptophan Operon. In this class, although it's in the notes, you don't need to know it for the exam, but using this table here, you can deconstruct whatever combination of regulatory operon that you were talking about. As an example, let me just quickly flip to the Tryptophan Operon so you see what I mean. The Tryptophan Operon is negative repressible. All right, you don't need to know for this exam, but if there is in the future, MCAT, genetics, microbiome, whatever, they go over some operon you've never heard of, and they tell you what combination of terms are used to describe it. Negative repressible, what does that mean? The phrase negative means a repressor protein is involved in some way. The phrase repressible means the goal is to turn off whatever operon you were talking about. Therefore, the only way those terms make sense together is that your goal is to attach a repressor to the operon to turn it off. That is what negative repressible means, is your goal is to attach a repressor to an operon. You would learn in that class or the test that involves a co-repressor. Okay? Again, you don't need to know any of that for this class. This is just to help you in the future. Yes, question?
So for the combination where they're both low lactose, isn't it pretty much the same end result?
Did you say if they were both low?
Both low lactose?
Yeah, if both are low, you can't get past step 1. So everything is off.
So it's the same user for both of those? So it would be the same user for both of those?
Yes, for both. As long as lactose is low, it doesn't matter.
Then you can't really use those.
All right, that's it. Whoever has a sign-in sheet, bring it up here.